Hello, I am Mercè Boada, the founder and Chief Medical Officer of Ace Alzheimer Center Barcelona. As a host, it's an absolute pleasure to introduce you to the first Global Research Summit. Since 1995, we've always had a clear purpose to bring Alzheimer diagnosis, treatment, research to a whole new level. Today, we are a renowned organization all over the world with a team of 100 professionals. Let me show you how we have got here. Since 1996, we have diagnosed around 30,000 people. We have collected 17,000 biological samples and led 138 clinical trials. Just now, as I am talking, we are doing 22 clinical trials targeting all phases of Alzheimer's disease and 9 national and international research projects, along with the highest level companies in the world. This allowed us to be heavily involved in the key scientific events of the last 2 decades. In fact, in 2020, even with the challenge of the pandemic, we have published more papers than ever before. Quite a journey, isn't it? But now it's time to set our sights on what's ahead with some passion and purpose from back in 1996. It's time to introduce you to the future of research. Welcome. Welcome to the Global Research Summit.
Good morning, good afternoon. I am Agustin Ruiz, the Scientific Director of Ace Alzheimer Center, Barcelona. I would like to welcome the audience to this second session of the Ace Global Research Summit 2021, organized by Ace Alzheimer Center and Grifols. As you know, today's session is going to focus on molecular biomarkers of neurodegeneration. We are very privileged to have a great panel of speakers, including Dr. Henrik Zetterberg and Dr. Michelle Mielke, who are going to talk about their developments related to this important field. First, please allow me to say a few words about the organization of this webinar. Each speaker is allotted 25 minutes time slot. After the talks, we will have 45 minutes for discussions and debate. Questions by the audience may be submitted through the chat during all the sessions and will be answered at the end during the discussion times. Also, questions through the microphone are also allowed if you ask for it previously in the chat. You can also send your thoughts through the social media as well. I also would like to take this opportunity to thank our partners for making this webinar possible and for their continued support. Lastly, I am delighted to introduce you to your chairwoman today of the session. Is Professor Charlotte Teunissen. She is a professor in neurochemistry in Amsterdam University Medical Centers, BU University. Her neurochemistry laboratory aims of developing biomarkers for neurodegenerative diseases, biomarkers that can be instrumental for improving biological diagnosis, therapeutical development, and monitoring of disease progression. Her current major disease topics are dementia and multiple sclerosis. Professor Teunissen, welcome, and please go ahead and start the session whenever you are ready. Thank you.
Good afternoon. Thank you very much, Agustin Ruiz, for the nice introduction of myself. I'm Charlotte Teunissen, professor in neurochemistry in Amsterdam UMC. It's my pleasure to introduce to you the seminars of this afternoon. First, we start with Henrik Zetterberg, and after that, there is a presentation of Michelle Mielke. The subject that they will deal with is very close to my heart and research interest. It's about body fluid biomarkers for neurological diseases and dementias especially. And this is an area where there is an increase and really enormous development, especially in the last years. It's very important, the biological biomarkers, the fluid-based biomarkers, they are now at the basis of the Alzheimer's diagnosis, of the biological diagnosis of Alzheimer's, and that means a paradigm shift. So nowadays we can define Alzheimer's disease by these fluid biomarkers without or independent of the clinical symptoms. So the clinical syndrome of dementia, it's a clinical paradigm and can be caused by multiple causes, but the concept of Alzheimer's disease is nowadays defined by the Alzheimer biomarkers, either by PET, but also by the fluid biomarkers, especially in CSF. And the very exciting developments of the past years are in the blood-based biomarkers, and that will also be addressed, especially by Michelle Melky this afternoon. And the blood-based biomarkers, they are really developing very quickly nowadays, and it's very exciting. And I do think that we can start to get ready for implementation, either in memory clinics, but also maybe in primary care settings. So it's very exciting and a very timely topic. The first presenter of this afternoon is Henrik Zetterberg. And Henrik Zetterberg is a professor in neurochemistry at the University of Gothenburg, Sweden, but also at the University College London in the UK. And he's a clinical chemist also at the Sahlgrenska University Hospital in Gothenburg in Sweden. He's the head of the Department of Psychiatry and Neurochemistry at the University of Gothenburg, and he leads the UK DRI Fluid Biomarker Laboratory at UCL in London. His main research focus and clinical interests are the fluid biomarkers for brain diseases, especially Alzheimer's disease. He has published more than 1,400 scientific articles and received numerous awards. So it's my pleasure to give the word to Henrik Zetterberg.
Thank you very much for inviting me to this meeting. My name is Henrik Zetterberg. I'm a professor of neurochemistry at University College London at University of Gothenburg. And I will give an update on the current status of CSF-based biomarkers for the diagnosis and prognosis of Alzheimer's disease. The last few years have seen extremely nice developments in this field, but one particular topic that has been in much focus has been blood biomarkers, but that you will hear from Michelle Mielke, who will speak after me. So I often start with this slide, which depicts a neuron and some other cell types of the brain. And the biomarkers we can measure now in cerebrospinal fluid with targeted assays include, for example, synaptic markers like SNAP25, synaptotagmin 1 and SV2A. And then we have a protein which is well known to many of you in the audience, Tau, which is a structural protein inside axons. And Tau can get phosphorylated, depicted here, p-Tau, and then it builds up in neurofibrillary tangles in Alzheimer's disease. So in Alzheimer's disease, there is an increase in CSF levels of both total and phosphorylated Tau. And that seems to be actually because the neurons are affected by amyloid plaque pathology here. And for amyloid plaque pathology, we can measure many different Ebitda forms and also amyloid precursor protein fragments. But those reflect more amyloidogenic and non-amyloidogenic APP processing, whereas Ebitda 42/40 ratio in CSF reflects plaque, the buildup of plaques in the brain. So Ebitda, Tau, and phospho-Tau are AD-specific biomarkers. Neurofilament light, on the other hand, is a biomarker of axonal injury and degeneration, irrespective of underlying cause. And that is, to me, a very important thing when I work here in the clinical neurochemistry lab. So if you have a neurodegenerative process in the brain, or if you have acute brain injury because of different diseases, then NFL levels will increase. It's a rather slow marker, so it increases with a maximum after about 2, 3, 4 weeks, and then it has a half-life of 2, 3 months approximately. In the neuron, there are many different proteins that are expressed at high levels and are released if neurons fall apart. The neuronal body is injured, sort of. And then we also have the dendritic tree of the neuron, which is reflected actually. The integrity of the dendritic tree is reflected by CSF neurogranin levels. In the brain, we also have astrocytes and microglia, and these cells secrete a lot of different proteins. These are just a few of those proteins that can be measured in cerebrospinal fluid and reflect astrocytic and microglial activation states. And then we have the blood-brain barrier, and for that barrier we have two biomarkers. The most well-established is the CSF serum albumin ratio, where we measure CSF concentration of albumin and the serum concentration of albumin. And the reasoning here is that albumin is made by the liver, and the albumin we can measure in CSF must have crossed the blood-brain barrier. And if this ratio is increased, the blood-brain barrier is leaky. Another more novel biomarker for the integrity of the blood-brain barrier, potentially a biomarker of the neurovascular unit, is soluble levels of Pdgf receptor beta. This is a protein highly expressed in parasites, and it's upregulated if the neurovascular unit is injured in different ways. Then, of course, there are other markers in the brain that, other pathologies in the brain that relate to neurodegenerative dementias. And one of the most exciting is TDP-43. The TDP-43 is a protein that can build up inclusions in some forms of frontotemporal dementia and ALS, but it's often seen also in other neurodegenerative diseases, often like a co-pathology in Alzheimer's disease, for example, and following traumatic brain injury. To date, we have had no good fluid biomarkers for TDP-43 pathology. The protein can be measured, but its level doesn't reflect the inclusions. And there are no good biomarkers for, there are no good imaging biomarkers for the pathology either. But just 2-3 months ago, an Italian group published a paper in Brain Communications that indicated that there is a possibility to detect seeds of misfolded TDP-43 in lumbar CSF of TDP-43 positive patients. And this is something we should pay attention to and follow up, I think. Very exciting. A similar situation we've had for alpha-synuclein, the protein that builds up the Levy bodies in alpha-synucleinopathies like Parkinson's disease and MSA and such disorders. CSF and blood levels of alpha-synuclein have been possible to measure, but again, they do not reflect the pathology. But now we have this similar assay as for TDP-43 that I mentioned. An assay where you take lumbar CSF, you spike in recombinant alpha-synuclein, and you check if there are seeds of misfolded alpha-synuclein that can initiate a fibrillation cascade in the sample, which you can follow with THT fluorescence. That assay is up and running in many laboratories across the globe, and it works. So we have the possibility of qualitatively address whether or not a patient has alpha-synuclein pathology in the brain, which is very exciting, I think. So now I will talk about how well the currently available CSF biomarkers reflect pathologies in the brain. And I will start with beta amyloid because there is so much data on this, and I think it's also it has become so clear how this works. So these are data here. All these are studies that have been meta-analyzed in the ALZ biomarker database of ALZ Forum. It's completely open. You can go in and check in all the different studies. We're updating this database as we speak now. We hope to have a new release during the Alzheimer's Association International Conference this year. All studies, this is a zoom in, show reduced levels of CSF Ebitda42 by approximately 50%. One is no change in this meta-analysis. There's one study standing out here, but that's the only study, and probably there was something wrong with that one. So CSF Ebitda42 levels in Alzheimer's disease are reduced, and that seems to depend on sequestration of the protein in amyloid plaques in the brain tissue. So the more plaques, the lower CSF level of beta amyloid. This is an old autopsy study with ventricular CSF, but now we can see this in amyloid PET studies also. So you see here, you can do amyloid PET, the redder the brain, the more amyloid in it, the higher the signal, I should say, of course. So here you see the amyloid PET signal. So high levels indicate amyloid pathology and low levels absence of amyloid pathology. And this is the CSF Ebitda42 concentration. So high concentrations are seen in amyloid negative people and low concentrations are seen in amyloid positive people. Then there are some discordant cases here. Longitudinal studies show that these individuals have an increased risk of moving into the amyloid PET positive quadrant. And this has been verified in different studies. This is just one study. Now I think there might be 20 or 30 or even more studies showing this. So it's a very concordant finding. But there is a small problem with CSF beta 42 as a biomarker of amyloid pathology. If you have a very diverse sample set. So, for example, if there is some neuroinflammatory conditions in the sample, neuroinflammation as such can bring down CSF beta 42 levels. Here are multiple sclerosis patients. These are healthy controls. These are multiple sclerosis patients, and here you see on the y-axis Ebitda 42 levels. And many of the multiple sclerosis patients would fulfill criteria according to cut points to be amyloid positive, which they obviously are not. I mean, these are rather young people in this study, and they most likely they have not undergone amyloid PET. But our interpretation of this was that there was something wrong here, something going on. But then if you look in the A and B panels, you see another phenomenon. Here you measure the shorter, more water-soluble Ebitda forms that are also secreted by all of us. And here you see controls, and then you see that the multiple sclerosis patients are lower also in these shorter forms, Ebitda 38, Ebitda 40. This does not happen in Alzheimer's. So in neuroinflammation, you can have a general downregulation of Ebitda peptides in the cerebrospinal fluid. And the same thing happens in normal pressure hydrocephalus. So here you see that Ebitda 38, 40, 42, soluble APP alpha and beta are concordantly lower in idiopathic normal pressure hydrocephalus patients. So there are other mechanisms that can bring down the APP and the Ebitda levels. And if you only look at Ebitda 42, you can mistake the patient for being amyloid positive, which would then result in your lab giving the patient a wrong diagnosis, sort of. The obvious explanation is to make a ratio. So CSF beta 42/40 corrects for this ratio, because in neuroinflammation and normal pressure hydrocephalus and potentially some other diseases, you have this concordant correlated reduction in both Ebitda 42 and 40, and the ratio stays the same. In Alzheimer's, you have a selective reduction in Ebitda 42 and the ratio is lowered. And here is a graph showing the effect of this ratio. So you have here CSF Ebitda 42 alone with a very nice AUC of 0.89 with this autoimmune assay. But if you add the ratio of Ebitda 42 to 40 or Ebitda 42 to 38, you get almost a complete concordance. And the reference standard here, I should say, for amyloid positivity is amyloid PET. So these are the same patients. Here's a mesoscale discovery assay that shows even better concordance of the ratio. And when we have done this now for quite many years in clinical neurochemistry, routine work here in the lab, use the ratio and Ebitda 42 alone also. If we look at the ratio of all samples we have analyzed during a time window of approximately 4 years, we see a bimodal distribution of the results. We can just look in the A panel here. So of course most are amyloid positive then according to the ratio, because most of our samples come from memory clinics and are investigations of suspected neurodegenerative disease. Then there is another group which is normal. And you see that the bimodal distribution, it's quite striking, and it's unusual to see this type of distribution for a clinical chemistry test. So here, to me, this means that you either age without depositing, and this is a normal distribution, or you age with depositing amyloid in the brain. And the lack of people in this valley between these two distributions suggests that you switch rather quickly from being a non-depositor to becoming a depositor. And I've wondered for quite many years, where are all these people? How quickly do you switch? And now we have just seen a data set from Sterling Johnson's team in Wisconsin, where we can address this. So here you see in their RAP cohort, these are cognitively normal people who age, some get some cognitive problems with time, of course. And here you see CSF Ebitda 42/40 ratio, repeated CSF samples over many years. And here you see the age distribution. So young people are normal, they don't deposit. When you age, there are people who dive down like this into the positive range of indicating amyloid deposition. So it looks like this is happening to many of us in the age range between 50 to 70, 75 years. Of course, there could be people who start to deposit here also. There are fewer observations here. But you see that people dive down rather quickly. It's a bit of a busy graph, but you see it still. So it doesn't take many years for the CSF Ebitda 40 ratio to become abnormal if you start to accumulate plaques on the brain. In this study, there was also amyloid PET. There is not a complete concordance so that all had both CSF and amyloid PET, but these are the data. Here you see people who are normal, they don't have amyloid on their brain, they don't deposit, and then you see that many age like this without getting amyloid, but then there are people who take off and cross the threshold for amyloid positivity. Then there is another thing here that I think, now this is a CSF talk, so I shouldn't talk about PET that much, but please see here how parallel these lines are. So this, to me, this means that when amyloid starts to deposit, the buildup is a chemical reaction. You can't live your life in a way that will affect the rate of amyloid accumulation, not that much at least. It's a chemical amyloid cascade, much like a prion disease, but slower. What I am interested in, and I think many of us in the audience and among the speakers are, is if one could, with lifestyle and or other interventions, affect when people start to deposit, when the amyloid cascade takes off. Perhaps we could see there that sleep and clearance of amyloid from the brain and such things could have an effect. The largest effect is ApoE here. So if you are ApoE4 positive, you have a much higher risk of getting early amyloid pathology, as many of us know. If you line up people according to when they cross when they are projected to cross the threshold of positivity, you see how small the variation gets. So amyloid buildup is not a variable process. It's a distinctive chemical process on the brain. When you start to deposit, it is a very distinct and non-variable process. So you see here, and if you then put all the people so that you know how close they are to onset of amyloid pathology, you can calculate for each individual how far from or to how long they've had amyloid on the brain, for example, time with amyloid. And then you see also that when you cross the threshold of amyloid positivity for Ebitda 40 ratio, a few years later you start to show amyloid PET signal, and then it's very distinct and non-variable. So it's to me, this has been extremely interesting to look at. So conclusions on Ebitda. There is a robust correlation to pathology. The biomarker is fully changed before symptoms appear. It does not change over time in symptomatic disease, at least not that much. It sort of dives down, and then we believe there is a plateau, but we are not certain on that. We need more longitudinal studies, of course. But it looks like that. And then we have CSF Ebitda 40 ratio. It's a robust, more robust plaque pathology marker than Ebitda 42 alone. As a laboratory physician, I think this is the easiest marker to interpret. It's easy to tell if a patient does or does not have amyloid on the brain, I think, and there is so much data supporting the statements we are giving. But from a clinical perspective, it's a difficult marker to interpret because you have to be super careful not to label a patient as an Alzheimer's disease patient if you can't link the symptoms to the pathology, because amyloid tests get positive in the preclinical stage. So if you have a person who comes to you who is depressed and show cognitive problems, you decide to examine him or her, you perhaps do a CSF analysis, and you find out that the ratio is low. Then you still have to figure out if this is the cause of the patient's symptoms, because this could be preclinical amyloid pathology that doesn't yet affect the patient's brain function, so that symptoms would appear. Perhaps the patient has depression with cognitive problems, and that is what is needed to be treated. And it would be a great shame if one would simply state to the patient that the cognitive symptoms, I've shown you have amyloid on the brain, you have Alzheimer's disease. It's early still, but it's Alzheimer's disease. So we have to be careful here, and we must not miss treatable causes of cognitive problems in amyloid positive people. And of course, we hope that there will also be treatments against amyloid positivity now, or treatments that could delay onset of the process. Perhaps they should be called primary prevention treatments then, but yeah. Good. There are certified reference methods and materials available, which is very good if you run a lab. And then there are fully automated clinical chemistry tests. They take around 15, 17, 20 minutes. So you could see your patient, interview him or her, perform a CSF sampling, get results back in half an hour or something like that, and then discuss the results with the patients. It's a quite nice setup now, and we these tests are available, quite available in clinical chemistry labs. Total tau is increased in Alzheimer's disease, as I mentioned earlier. Many studies. These are also biomarker data, database data, and then but there are problems then with total tau, and one of the obvious problems is that this marker, which we thought this was a neurodegeneration marker from the start, since it's an axonal protein, and when axons degenerate, we imagine that they would release their tau content. But if you look across neurodegenerative diseases, this is an old study from Ito et al. In Japan. You see that for FTD patients and CBD patients and PSP patients, they don't have total tau increase. This means that total tau is not a neurodegeneration marker. And thanks to hard work by Matthias Jucker, Randy Bateman, and other people in their teams and also other groups, it now seems like amyloid is inducing tau secretion from neurons, and that's why we see this result. So tau is an amyloid marker. At least perhaps this is an oversimplification and a little bit confusing also, but I start to think about it like this. Also phospho-tau then is an amyloid marker. One shouldn't state like this, I think. I think it's better to say like this. Total tau and phospho-tau are markers of amyloid-induced tau pathophysiology or something, and this will eventually correlate with tangle pathology and neurodegeneration, but they are not direct markers of those two processes. That's how I think we should talk about these markers. You see here that phospho-tau is also AD specific. Good. These are just data from our lab. It's showing the same thing. The higher the total or phospho-tau level, the faster disease progression. This is progression to death. So it's sort of a disease intensity marker in Alzheimer's disease also. Here's phospho-tau concordantly increased in Alzheimer's disease, but weak correlation with pathology, which I think resonates well with what I just said, that it's a marker of tau phosphorylation, which might be then induced by amyloid. Neurons that are affected by amyloid will eventually develop neurofibrillary tangles. So then you might see a weak correlation with phospho-tau, and it might be that phospho-tau then is a predictive marker rather than a direct marker. Here you see an early tau PET study. The correlations are a bit better now, but the correlation between tau PET imaging signals and phospho-tau levels, they appear in the dementia stage, but they are absent in early disease stages, or weaker at least. So CSF total tau and phospho-tau are increased in Alzheimer's disease, but not in several other tauopathies. CSF tau is normal in non-AD neurodegeneration. That is, it is not a general neurodegeneration marker. There's a big correlation with pathology. CSF tau concentration correlates with rapid disease progression. Increased tau secretion could be a disease intensity marker, and then this could be very useful in clinical trials of Alzheimer's disease. Increased total tau and could then be, and phospho-tau could be a marker of a neuronal response to Ebitda pathology. I think this is almost proven now. The other non-AD tauopathies, why can't we detect the tau signal in those, like frontotemporal dementia and PSP? Well, perhaps we measure the wrong form of tau, or this is a process that occurs in the tissue and is not reflected in the fluid. So for ATN criteria, amyloid tau and neurodegeneration for patients with suspected Alzheimer's disease, we can use CSF Ebitda 42, especially if it's in a ratio with Ebitda 40. Phospho-tau is a predictive marker, but not a direct marker of the T criterion. And total tau is a predictive marker of neurodegeneration, but not a direct marker. I should say that total tau increases after stroke and traumatic brain injury and Creutzfeldt-Jakob disease, but in other neurodegenerative diseases, the increase seems to be AD specific. Here you see the candidate biomarker for the N criterion, neurofilament light. This is it fulfills many criteria for being a neurodegeneration marker. If you have high levels, you lose cognition more fast, you get larger ventricular volumes, and you lose brain tissue. It is increased in all neurodegenerative diseases except Parkinson's disease. It reflects disease intensity in those diseases where we have effective treatments. So this is multiple sclerosis, where you have axonal injury, and then you treat your patients with an effective drug, in this case natalizumab. The levels go down to control-like levels. This is what we need to see in Alzheimer's and other neurodegenerative diseases. It works in spinal muscular atrophy, which is a childhood neurodegenerative disease that can be disease modified by injections in the lumbar sac of an antisense oligonucleotide that stabilizes a transcript for the deficient protein. And when you start to inject the drug, neurofilament light levels go down. The children survive. You see their ages in months here. The crosses here represent normal children who did not have a neurodegenerative disease, so these we regard as the normal levels. You treat the patient with a disease-modifying treatment, the biomarker normalizes, the patient gets better. I think I skipped this. This is then the conclusion, not the conclusion, but the next step of all this. Can these biomarkers be measured in blood? And this you will hear much more about from Michel Mielke and all the exciting developments that have happened here. And I think we will discuss this also. And Charlotte Heunissen, who is chairing this session, we will lead that discussion. My lab in Gothenburg and my lab in London, and then I would like to thank you for your attention.
Thank you, Dr. Zetterberg. That was very interesting. The second lecture of today is given by Michelle Mielke. It's my pleasure to announce her. Her bio is as follows. She is currently professor at epidemiology and professor of neurology at the Mayo Clinic in the USA. She's also the Associate Chair of Faculty Development and Academic Affairs in the Department of Quantitative Health Sciences. Dr. Mielke works as a translational epidemiologist to further understanding of the etiology and epidemiology of neurodegenerative diseases. One focus of her research is the identification of fluid biomarkers for the diagnosis, prediction, and progression of Alzheimer's disease and other neurodegenerative diseases. Another focus of Dr. Mielke's research is on understanding sex and gender differences in the development and progression of Alzheimer's disease and of other aging-related conditions. She directs the Mayo Clinic Specialized Centers of Research Excellence on Sex Differences. Let me give the word to Professor Mielke, and she will present you and give you an update of the current state of blood-based biomarkers for diagnosis and prognosis of Alzheimer's disease. Please, Dr. Mielke, please take the floor.
Hi, my name is Michelle Mielke, and I'm from the Mayo Clinic in Rochester, Minnesota. Today, I will be discussing the current status of blood-based biomarkers for the diagnosis and prognosis of Alzheimer's disease. First, I will start by giving a very brief background with regards to the ATN research framework from which the biomarkers will be built on that I will discuss. Then I will talk about the current status of biomarkers of amyloid beta, followed by phosphorylated tau, and then markers of neurodegeneration, including both total tau and neurofilament light. So the ATN research framework. Historically, a diagnosis of Alzheimer's disease, or AD, was based on clinical symptoms. This was problematic because symptoms can be due to multiple pathologies, and some individuals being enrolled in clinical trials for AD did not have elevated brain amyloid. CSF and imaging biomarkers of amyloid and tau, the two key pathologies of Alzheimer's disease, became available for research purposes, and CSF has been used for the clinical diagnosis of AD for several years. As a result, the NIA and Alzheimer's Association collated a panel of experts that developed a framework for a biological definition of Alzheimer's disease that could be used when recruiting for clinical studies or to better understand underlying disease mechanisms and progression. The grouping classification was for amyloid and tau, which are the key pathologies of Alzheimer's disease. In addition, neurodegeneration is in parentheses because it is nonspecific and occurs across neurological diseases. Initially, imaging and CSF markers were proposed to ascertain the presence of amyloid, tau, and neurodegeneration, or ATN, but the framework left open the possibility that additional biomarkers could be added. And with recent advances in technology, we now have the potential to add blood biomarkers of amyloid, tau, and neurodegeneration. And of course, blood measures are certainly less invasive and costly and more feasible from a population-based and primary care perspective compared to CSF and neuroimaging. So first I will talk about blood amyloid beta. Blood amyloid measures have actually been examined for the past couple decades. However, there's been a lot of conflict between studies, for example, whether elevated blood amyloid beta levels or decreased amyloid beta levels were a risk factor. And part of the reason for this conflict was that there was a lot of variability in the amyloid measures. So it was not feasible to predict progression from an individualistic standpoint. This started changing in 2017 when two articles were published around the same time from two different groups, both using a mass spectrometry-based approach. The OVID paper was published by colleagues in Washington University in St. Louis, Missouri. They found a high accuracy of the amyloid beta 42:40 ratio in separating those individuals who were amyloid positive from those individuals who were amyloid negative, with an area under the curve of almost 0.9. The ratio of 42:40 was lower among those individuals that were amyloid positive. The other study included groups from Japan and Australia. They also found a high accuracy of the amyloid beta 42:40 ratio for predicting an amyloid PET. Notably, in this latter study, the opposite ratio, 40:42, was used, but the results are really in the same direction, again indicating that low plasma amyloid beta 42 or the amyloid beta 42:40 ratio is indicative of brain pathology. Since these publications, there have been many other methods and studies published measuring blood amyloid beta, typically in the plasma. There are still a lot of questions as to what method is preferable for a given use, for example, at the population level or more for a specialty neurology clinic. However, this past year, C2N Diagnostics developed the first plasma amyloid beta test for clinical use, called Preclivity AD. This is a mass spec-based approach and uses an amyloid probability score, or APS, that incorporates a proprietary algorithm, including information on the plasma amyloid beta 42:40 ratio, ApoE4 genotype, and age. Patients can have a score of low, intermediate, or high probability of having amyloid pathology, with intermediate results indicating indeterminate results. The current cost is $1,250 US dollars and is considered out of pocket. So it's not covered by Medicare or health insurances right now in the US. The blood is sent to Washington University and the results are expected in roughly 10 days. Note that this is available in the United States, with the exception of 3 states, and also in Puerto Rico. However, I'm unsure where things currently stand for access in Europe as well as other countries outside of Europe. So in terms of thinking of next steps, going forward, I think there is a need to compare the pros and cons and utility of the different assays for contexts of use, including the mass spec assay currently available clinically, as well as other ELISA-based or Simoa assays. In addition, longitudinal studies with serial blood measures for more diverse and larger studies are needed. This is important to understand how these markers look in the general population, which is highly heterogeneous, as well as how to best interpret these markers. Next, I will focus on plasma phosphorylated tau. In 2018, we, in collaboration with Jeff Dage and others at Eli Lilly, published a paper showing the potential of plasma p-tau 181. We found significantly elevated levels of plasma p-tau 181 in those with elevated brain amyloid based on amyloid PET compared to those without. So within each clinical diagnosis, cognitively unimpaired or MCI, those who were amyloid positive had significantly higher levels. Further, we found that plasma p-tau 181 correlated both with amyloid and tau PET. So among cognitively unimpaired individuals, higher levels of plasma p-tau 181 correlated with amyloid PET, and then among MCI and AD patients, high levels of p-tau 181 correlated with both amyloid and tau PET. In addition, we showed that plasma p-tau was as good or better than age NAPOE for predicting elevated brain amyloid. So these results were conducted in the Mayo Clinic Study of Aging, which is a population-based study of Olmsted County, Minnesota residents. Since that initial paper, there has been just a mass explosion of papers with similar findings across a large number of clinical and neuropathological cohorts. Only 2 of which I have cited here, but there are probably at least 20 papers out now showing the potential utility of plasma p-tau 181 and other species. These studies have consistently shown that plasma p-tau 181 is elevated in those who are amyloid positive compared to amyloid negative, associated with increasing Braak stage, that high levels of plasma p-tau are associated with an increased risk of dementia, specifically Alzheimer's disease. And importantly, that these p-tau blood markers are specific to the 3R4R tauopathy, which is observed in Alzheimer's disease, as compared to other dementias such as frontotemporal dementia, which is either only a 3R or a 4R. Thus, this marker is specific to Alzheimer's disease. So these studies have been just extremely promising, but there are some important next steps before they can be considered for clinical use. For example, almost all the studies to date of plasma p-tau have been focused on clinical populations. As I mentioned with amyloid, it is important to examine the utility of plasma p-tau in the general population and to decide whether there are factors that can affect the interpretation of the levels. There is also a need to identify cut points for clinical use. Lastly, although I initially showed data on plasma p-tau 181, there are additional p-tau isoforms as well as assays, and these need to be compared to determine how they change in relation to each other and which might be most useful in different contexts. So to start looking at this in the general population, we started examining plasma p-tau 181 and p-tau 217 measured on mesoscale discovery platform in the Mayo Clinic Study of Aging, including over 1,000 non-demented individuals from the community. Here we show that plasma p-tau 181 and p-tau 217 levels increase with age, particularly starting around the age of 65. Notably, however, when we stratify by amyloid PET status, the increase with age is only in those with elevated brain amyloid. This finding is important because it indicates, first, that these p-tau markers are solely related to Alzheimer's pathology, and second, the small increase among amyloid negative individuals makes it easier to develop reference ranges. In other work, we have been trying to understand what factors affect the interpretation of levels of the blood markers. This specific graph focuses on plasma p-tau 181 and 217, but we are also working on analyses, also focusing on other blood-based biomarkers, including plasma amyloid beta 42, neurofilament light chain, and total tau. There are a couple things that I want to point out in this figure. So p-tau 181 is on the left and 217 is on the right. And for the most part, the associations are quite similar across the different p-tau markers. At the bottom, we can see the difference between amyloid beta or amyloid PET positive individuals and amyloid PET negative individuals. So those individuals that are amyloid PET positive have at least 0.5 standard deviation higher level of p-tau 181, and even more than that for p-tau 217, compared to those who are amyloid negative. However, there are also some significant effects of other factors as well. So the difference between those individuals that are amyloid positive and amyloid negative is essentially the same as those individuals that have chronic kidney disease or CKD compared to those that don't. And even when we stratify and only look at the effect of CKD among amyloid negative individuals, there still is a very large impact of CKD on increasing p-tau levels. A couple other factors that also appear to affect levels are stroke and myocardial infarction as well, with almost a half standard deviation increase for those who have these conditions compared to those that don't. So as I mentioned, this information is important for developing reference ranges, but I think it's also important to consider from a health disparity standpoint, particularly with the examination of differences by race ethnicity. So, for example, there have been several studies more recently that have been comparing levels of plasma biomarkers as well as CSF and other biomarkers between, for instance, African Americans and whites. And African Americans have a higher prevalence of CKD, chronic kidney disease, myocardial infarction, and stroke, all of which affect plasma p-tau levels. So therefore, we need to consider the possibility in the future that racial differences in blood-based biomarkers could be actually due to the difference in prevalence of these conditions, all of which are higher in Blacks than whites. And so it could be the conditions that are resulting in a race ethnic difference, and these conditions need to be considered in the future when comparing results between populations. In terms of predicting elevated brain amyloid using continuous measures of plasma p-tau, we do find that p-tau 217 is significantly better than p-tau 181 for predicting abnormal amyloid PET. In addition, both p-tau measures are as good or better than age, sex, and ApoE alone. However, for predicting abnormal tau PET, there really was no difference between the p-tau measures, and the p-tau measures alone actually performed a bit worse than the predictive value of age, sex, and ApoE. So we think that these findings further indicate that plasma p-tau, especially 217, change early in the disease process, possibly even before tau pathology can be measured with tau PET. Next, to build on our findings of factors that affect the levels of these p-tau measures, we have tried to develop some initial cut points for each marker, which of course are critical for clinical use. Using 2 standard deviations above the mean among amyloid PET participants, we developed cut points for each p-tau measure. In addition, because I just showed that chronic kidney disease, stroke, and myocardial infarction are associated with higher p-tau levels, even among those who were amyloid negative, we excluded individuals with these conditions and evaluated the predictive values of these markers. As you can see, the cut point really did not change for p-tau 217, 0.25 versus 0.26. But there was quite a drastic change in cut points for p-tau 181, going from 1.57 after excluding individuals to 1.75, including these individuals, which may suggest that there's just more variability in p-tau 181. When we look at these cut points for predicting amyloid PET as well as abnormal tau PET, we see that both p-tau 181 and 217 have very high specificity. In addition, the positive predictive value, or PPV, is quite high for amyloid PET. Using either cut point, it's higher for p-tau 217 than it is for p-tau 181. Negative predictive value of these markers for amyloid PET is a little bit lower, but still quite good. And then for tau PET, the negative predictive value is quite high. Notably, sensitivity of these markers for amyloid PET and tau PET are quite low. In additional work, as I mentioned, there are different plasma p-tau species. So here we did a direct comparison of p-tau species, 181 and 217 using the mesoscale discovery platform, and 181 and 231 measured using the Simoa platform. For all species, you can see that plasma p-tau levels are higher for those with elevated brain amyloid, regardless of clinical diagnosis, compared to those without. However, the differences between amyloid positive and amyloid negative among cognitively unimpaired was higher for those with, or for p-tau 217. For the MCI comparison of amyloid positive and amyloid negative, the different species and platforms were much more similar. When we compared each of these p-tau markers for predicting amyloid and tau PET, there was actually very little difference though. So in summary, plasma p-tau consistently correlates with both amyloid and tau PET, and may even be an earlier marker to assess AD pathophysiology than blood amyloid, especially as it seems to be a better predictor of amyloid PET. I think there is still a lot of work that needs to be done to understand the different p-tau species. So in our initial analyses with a much larger sample size of over 1,000, we did show that 217 was a significantly better predictor of amyloid PET than 181. However, in the results I just showed you with comparison across platforms and species and a smaller sample size of 200, we didn't see much of a difference. So certainly much more work is needed in larger sample sizes and also community-based samples. In terms of next steps, as with plasma amyloid, longitudinal studies with serial samples are needed to understand how the measures change over time in relation to the clinical progression of the disease, and also how the p-tau species change in relation to each other over time. Additionally, in terms of clinical use, cut points will be important, and so there will be much additional work that is needed to develop these cut points and to standardize them across populations. Lastly, I'll briefly touch on two markers of neurodegeneration, the leading candidates of blood, of which are total tau and neurofilament light chain. So we and others, including the Framingham study, Alzheimer's Disease Neuroimaging Initiative, and several other cohorts, have shown that high levels of plasma total tau are associated with greater memory decline and also greater risk of MCI. So both the middle and highest tau here are associated with increased risk of MCI. However, across these studies, these associations are independent of amyloid PET, indicating that plasma total tau is more of a non-specific marker of neurodegeneration. There are also several studies that suggest that blood measures, either plasma or serum, of neurofilament light is a non-specific marker of neurodegeneration and also independent of brain amyloid levels. For example, in this first paper using ADNI participants, NFL is associated with global and domain-specific cognitive decline, increase in ventricular volume, and hippocampal volume loss. Importantly, blood NFL strongly correlates with CSF NFL. And when we compare blood levels of NFL to CSF levels of NFL in relation to various neuroimaging measures as well as cognition, the results are essentially the same. indicating that blood measures of NFL are as good an indicator of cognitive decline and neurodegeneration measures as our CSF measures. So given that both total tau and neurofilament light are nonspecific markers of neurodegeneration, we wanted to compare the two with regards to cross-sectional and longitudinal measures of global and domain-specific cognitive decline and different neuroimaging parameters to try to discern when one marker might be more useful than another. For example, if a patient does come into the clinic, is it important to measure both markers, or is one better than the other? In terms of cognition, when we looked at baseline, there was really no difference between plasma NFL or total tau in relation to cognitive impairment. However, longitudinally, higher levels of each standard deviation increase in NFL was more significantly associated with cognitive decline compared to a standard deviation increase in total tau. With regards to neuroimaging measures, again, when we looked cross-sectionally, there was really very little difference between NFL and t-tau for any of our neuroimaging measures. However, longitudinally, NFL was more strongly associated with declines in temporal thickness, in hippocampal volume, as well as corpus callosum fractional anisotropy, a measure of white matter integrity. In further analyses, we also replicated this work using the Alzheimer's disease neuroimaging participants, and the results were essentially the same, indicating that prognostically, NFL is a better marker and a more stronger predictor of cognitive decline and neuroimaging changes than total tau. So in terms of blood total tau and NFL, I think these results suggest that plasma NFL has better utility as a prognostic marker. However, although I didn't show you some of the data, when we do stratify by amyloid PET status, there is a little bit of value added with plasma p-tau among those that are amyloid positive. So it is possible plasma total tau can add some diagnostic benefit cross-sectionally, but again, prognostically does not appear to add any value, at least in the two studies that I've showed you. Again, going forward, as with the other plasma markers, longitudinal studies with serial samples are needed, particularly to compare the rate of change in these biomarkers in relation to progression of clinical symptoms and brain pathology. So in summary, I've very briefly shown you some of the new blood-based biomarkers. And it's very clear that over the past couple of years, the field is rapidly expanding in terms of biomarkers of amyloid, phosphorylated tau, and neurodegeneration, and that these biomarkers are moving towards clinical use. However, as I've also highlighted, additional work will be needed to standardize the assays and compare platforms, and to understand what will best be used in the community settings. There is certainly a need to compare these markers in the general community and more diverse samples as opposed to neurology or specialty clinics. This will be particularly important in considering how to use these biomarkers, whether they should be used more diagnostically for specialty clinic purposes, or whether they can be used in primary care where individuals first present with cognitive symptoms. Thank you.
Thank you very much, Michelle Melky and Henrik Zetterberg. Now it's time for discussion. And I observe that we already see a lot of questions within the chat. So I would like to ask Michelle and Henrik to enter the floor also. Well, there they are. And also Agustin and Luis. And I'd like to start with the first question. Chronological order. So Isabel Rodriguez, Lucas Prijs, he asked if tau is an amyloid-induced marker, why do we not typically see tau pathology in DLB, even though amyloid accumulation is seen quite often? Yeah, so maybe you have to rephrase whether tau is an amyloid marker or amyloid-induced marker.
Sorry. Yes, that's a great question. It's to me, it looks like this, that some DLB patients, if you look at many DLB patients, they often are in between the control group and the AD group. That's my experience, at least. So to me, it looks like there is a little bit of amyloid effect on neurons in most cases of DLB. But of course, there is a combination here of alpha-synuclein pathology and amyloid pathology, and potentially that makes the picture a little bit more complicated. So I think there is still some kind of relationship between tau and amyloid also in DLB. I do not know if you would like to comment on that also from your clinical work, Charlotte.
Now from our CSF biomarker work, because Lisa Vermunt, she has dived very deeply into an Olink proteomic study that we performed, and she compared the DLB patients with amyloid pathology, for example, with AD patients. And there we see very distinctive patterns. So I do think that the people of DLB with amyloid pathology and also tau pathology, they are different. They are not just DLB plus Alzheimer's disease, but there is something, really specific. Indeed, probably due to the interaction between different proteins and other processes.
Mm-hmm. That is something we should make use of in the research, I think.
Yeah.
If there is something special with the amyloid plaques of DLB that differentiates them from Alzheimer's. Yep, very interesting.
You should be able to see that also if you do studies. Yes, let's continue because we have a lot of questions. Matthias Schreuter, I think, gives you compliments. Very interesting thought. Thanks. Can CSF biomarkers or their patterns differentiate between clinical subtypes of Alzheimer's disease, for example, the Alzheimer's dementia with memory dysfunction or logopenic PPA or posterior cortical atrophy?
Yeah, the short answer there is no, unfortunately not. And this is exactly where we can make use of molecular imaging. So it's so PCA, AD, and PPA look rather similar from a CSF AD biomarker perspective.
Okay, thank you. And a question from Merce Boada, the chair of this meeting. Again, also a compliment. And then the question is, can you prioritize all the CSF biomarkers focused on vascular burden on microglia activation or in general gliosis? So the question pertains to, are there biomarkers for different mechanisms that we are interested in?
Yep. That is also very important and a hot topic for further research. If you have cerebrovascular disease, there is often oligodendrocyte deficiencies and myelin deficiencies, and then you also get this neurofilament light increase, which might be a little bit more pronounced than in Alzheimer only. But it's definitely not a specific marker of cerebrovascular disease. There are exciting biomarkers coming up that seem to reflect the function or integrity of the neurovascular unit. And one very interesting molecule is the parasitic proteins, Pdgf receptor beta, that has a soluble form which is secreted into the CSF. So blood-brain barrier dysfunction is typical, of course, of cerebrovascular disease. And if you have then biomarker evidence of blood-brain barrier dysfunction through the CSF serum ratio, albumin ratio, or increased soluble Pdgf receptor beta, that could potentially indicate this cerebrovascular disease. But it would be wonderful if we could have better markers of cerebrovascular disease, or more distinct. There are studies on extracellular matrix remodeling proteins that seem to be activated if you have slight hypoxia in the tissue, and those we could look into further. And then if we think about other markers, for example, it was a question regarding microglial activation marker. The best protein biomarker I'm aware of is soluble TREM2 because it has a CNS selective expression. Microglia selective expression, sorry. So the microglia are are often hard to differentiate from astrocytic activation, but soluble TREM2, if you look, for example, in the Human Protein Atlas, has microglia specific expression. It's also expressed in peripheral macrophages, so blood soluble TREM2 levels do not tell anything about what's going on in the brain. And then we have the astrocytes, and the astrocytes has a very nice and well-established biomarker, which is glial fibrillary acidic protein, GFAP. That's a traditional marker in rodent models of CNS diseases. We often use staining of GFAP to look at astrocytic activation, and activated astrocytes also secrete GFAP. And GFAP works really well in blood. It actually looks better as a marker of astrocytic activation if you measure it in blood than in CSF, which is interesting. And I wonder if that has any relationship to the astrocytic end feet in the neurovascular unit, so that could provide an effective clearance pathway into the bloodstream, perhaps better than into the CSF. So there are a number of markers that are not just related to amyloid, tau, and neurodegeneration that will be interesting to learn more about through studies.
Yeah. And what do you think of, that's my own question, oxidative stress biomarkers?
Yep. When I started in this field, Domenico Pratico did a lot of work on lipids, oxidized lipids, exactly. I do not know what happened with that. It looked really promising, but I do not know really. It didn't take off completely.
No, I don't think they were really specific for Alzheimer's disease. They are rather oxidative stress biomarkers that you can also measure in other diseases like multiple sclerosis, for example. Mm. So I don't think they have the full specificity. But
perhaps that's not expected either.
No, yeah, exactly. And if you measure it in CSF, it can still be a good biomarker. Mm. Okay, let's move on to the next question. Amanda Cano, she has a lot of questions, but they are for Michelle. Do you think that plasma p-tau 217 is good enough to discriminate between Alzheimer's disease from other neurodegenerative diseases in general?
Yes, I do. I mean, I think both p-tau 181 and p-tau 217 in the blood are very specific for Alzheimer's disease.
Yeah. No, I can agree with that.
All right.
Yeah.
But could I ask a question there quickly? These effects upon myocardial infarction and such things, what do you think that represents?
That is a great question, and we've had a lot of discussion around that. Also about trying to recruit some patients with myocardial infarction and just follow them and try and understand that. I don't know. I mean, I think with chronic kidney disease, it's a lot clearer that it's more the clearance of the proteins. And then with stroke, of course, there could be a lot of effects as well, but I don't know with myocardial infarction.
Continuing with this line, Michelle, I wonder also if you wanted to add, beyond the morbidities affecting to the measurement of the tau in plasma, do you have any insight on matrix effect in the measurements? Any protein impacting the measurement coming from plasma? Yeah.
That is a great question, and that is something that we haven't looked at. We took advantage of our epidemiological study where we could go back through the medical records and really accurately diagnose all these various conditions. But I wonder, Charlotte, to send it back to you, because you've done a lot in terms of standardization. Have you looked at any of these protein effects?
Do you mean in terms of a pre-analytical variation?
Or the matrix itself, for example, hematocrit or any other protein affecting the measurement?
Yeah, I'm not aware of other proteins that affect the measurements, yeah, well, we get rid of the matrix because we have now more and more sensitive assays. So we think it's gone if there would be any effect. but probably there is, because we see some differences if you collect heparin instead of EDTA plasma, and that's throughout all biomarkers that we measure. So I don't think that's a specific mechanism pertaining to any of these biomarkers, but rather a more general mechanism, and that could be the matrix that they are in. Now, it could likewise be that it's related to some cellular release, of course, and that affects all biomarkers, but my current hypothesis is that it's some kind of matrix effect. The next question, also very interesting one. What do you think is the future for plasma exosomes as novel biomarker carriers for Alzheimer's disease and other neurodegenerative diseases? That's a very broad question. Maybe we focus on Alzheimer's disease, but exosomes in general. And either of you can answer it, because it could be either in CSF or in blood. We made this distinction this afternoon, CSF for Henrik and plasma for Michelle. But who wants?
So I mean, I think there's just a lot of interesting research coming out in terms of examining the extracellular vesicles, not only for amyloid tau neurodegeneration markers, but for a whole host of other measures that may be related to Alzheimer's disease as well. I think, one of the limitations with the extracellular vesicles is that there are a lot of methods that aren't necessarily standardized right now. So just to kind of give a shout out in terms of the biofluid-based biomarker group, which Henrik and Charlotte chair, Robert Risman is leading an effort on this in terms of trying to prioritize and standardize some of these methods, because I think that's the biggest limitation right now in terms of trying to validate these markers across studies.
And I could just add that perhaps for phospho-tau and neurofilament light, we might not need the methods, the exosomes when we are making blood assays, because it looks so good. But for the biomarkers where there is peripheral expression that really influences the blood levels in a way, making the blood levels useless when it comes to interpreting what's going on in the brain, perhaps there we could see a benefit of isolating neuronally derived exosomes, measuring those proteins, and by that approach get a CNS-specific signal. That would be very exciting if that was the case. And perhaps there is also, perhaps that could be a way through which we could learn more about or get blood biomarkers for synaptic dysfunction, microglial activation, CNS inflammatory processes, perhaps also dysregulated RNA molecules and other species that we don't look at that much or that gives a too noisy signal in the complex blood samples.
Oh, great. You answered already one other question, or you started to answer another question. What about RNA as possible biomarkers?
Yeah, okay, good. There are some studies, and I guess that they will become, there will be more on where RNA molecules are sequenced and you try to find profiles of RNA expression. It's a bit hard to find a consistent pattern that has been replicated across studies, but within studies there are signals that could be explored further, and perhaps if this could be done in exosomes, that could also potentially give us clues on dysregulated pathways that we could examine further. So I would say interesting, but still in need of further work and replication studies.
Yeah, and if I may add to that, similar as for the exosomes, I think there's a lot of standardization work needed still, especially if you look into the microRNAs into the CSF. And I think the concentrations are very low and difficult to measure, or it's maybe the isolation procedure that we should work on. So it's a method that still needs some further development and maturation. Now I have to jump back. As the question of Javier Morato, as almost all amyloid beta peptides traffic in plasma bound to albumin, does classical ELISA detect with the same efficiency both bound and unbound amyloid beta peptides? This would explain why mass spectrometry gives better results, isn't it? Who wants to answer? Is there, what do we detect in blood? That's the question.
Should I start and then perhaps you can fill in? I think this is, I think the question is on the spot actually. So if we, I mean, most of the plasma Ebitda will be bound to proteins, that's for sure. And if you dilute sample with a little bit of detergent, you don't get a linear dilution curve. You will mobilize amyloid from the matrix, as Charlotte just said. So it's very important to make the amyloid available for measurement. And if you just do a crude ELISA on plasma, you might get a signal, but it will not reflect the real concentration of beta amyloid, neither 40 nor 42. But with the SIMO assays and some of the other very sensitive assays, you can dilute the sample with some detergent and get most of the Ebitda in solution. But the dilution linearity is still not perfect, indicating that we do not measure all of it. With the mass spec-based assays, they are also dependent on an immunoenrichment of the proteins if you do the plasma Ebitda assays. So you start with anti-Ebitda antibodies onto some beads, perhaps magnetic beads. You extract the amyloid from the sample. You elute the extracted amyloid, and then with the C2N method, you digest the sample and look at the C-terminal truncation pattern and make these ratios, Ebitda 42 to 40 or Ebitda 42 to 38. In the method we have developed, we do not digest the sample. We immunoenrich for Ebitda, and then we measure intact Ebitda 1 to 37, 1 to 38, 1 to 40, and 1 to 42. In the blood, there is another confounder, and that is that there is enzymatic activity that can digest Ebitda. So if you take a whole blood sample, you will and incubate it at the bench, you will get a cleavage at amino acid 20 of the Ebitda peptide, and that will affect both Ebitda 40 and 42 signals if you measure the intact protein, which you do in most of the sandwich ELISAs and with the mass spec-based method we have developed. But the C2N method would be less sensitive to that. So perhaps this is explaining why it works so well, actually. Otherwise, I've thought that the Ebitda 42-40 ratio from a clinical lab perspective is a nightmare biomarker, because the ratio is changed so little, 10, 12, 14% in the plasma if you are the reduction is 10 to 15% in plasma if you're amyloid positive, compared with 50% in the CSF. And keeping such an assay stable week by week in a lab, that's tough. And yeah, I think that can give a bit of headache if you're responsible for that lab.
The artifact that you mentioned, but then we should see more differential effects of leaving the samples on the bench when you measure different assays, and that's not really what we see in a systematic evaluation of the pre-analytical factors. So we compared at least six different assays, also mass spec and immune assays, and left the amyloid sample on or the blood sample on the bench. And then we saw a reduction in amyloid beta 42 levels that were similar across all assays. So I wonder how we can align those results.
I think it's like this. The effect is clearest in whole blood, and it also looks like EDTA can inhibit that effect a little bit. So perhaps the proteases are divalent cation dependent. Yeah, so exactly. But in vivo it definitely happens. So you get some Ebitda 1 to 20 and 21 to 40 in the blood. Those are not present in CSF. At least we have not been able to detect them.
Yeah.
But it's but when we talk about biomarkers in clinical practice, one concept is robustness. And that's a clinical chemistry, very vague concept, but essentially it means if a biomarker is robust, you can introduce a little bit of variation or bias without losing clinical without losing diagnostic accuracy. And that if you introduce a little bit of bias in Ebitda 42-40 measurements, you immediately lose accuracy. So it's a very perhaps that's not the right English word, but volatile. It's not a robust marker. One can probably get it robust, and it looks like then C2N and perhaps some other labs have been able to start to do that. But the reason for why age and ApoE is included in the algorithms is to try to make the biomarker test a little bit more robust. But it's important to remember that age and ApoE in combination with the Ebitda to 40 ratio, then the measure stops being a biomarker, because you're born with your genotype and you age irrespective of whether or not you get amyloid. So it's a bit of I think it muddies the waters a bit to mix these. It works then it becomes a prediction tool for group level data.
But I think including age and ApoE in the model is also a way of taking care of confounding factors, not to see them as independent biomarkers. That's how I see it.
Mm-hmm.
Yeah, I have a question also related to this, because actually the percentage of ApoE carriers and the age variance between different data sets is also impacting to the modeling of the statistical modeling to measure the area under the curve. And this is something that I wanted to ask to all of you, if to compare different techniques. I think the optimal comparison is to use the same techniques without covariates in the same data set. Otherwise, you will be impacted about the information contained or provided by age or a percentage of ApoE carriers, for example.
Yeah, no, I completely agree with that. And I think, a lot of times when we're trying to maybe do meta-analyses or summary of the review of the literature, we report maybe the assay, but not necessarily the characteristics of the study. And so I really think that is critical going forward. Certainly ApoE has a large effect, as does age. And, I like to think about, thinking in our Alzheimer's Disease Research Center, there's a big interest on different types of Alzheimer's cases. And so those that tend to be referred may have a little bit more executive function, or maybe they're younger. And so we always have a difficulty when we're using our community-based sample, which it tends to be a little bit older, and then we need 80 cases, and our 80 cases are younger and a little bit different. And those don't typically come to light in the paper, but that's certainly an issue.
Mm-hmm. Yeah, thank you. Very elucidating also. I have a very interesting question of Maria Solaguren Basoa. Now, I can't, I'm sorry if I pronounce it wrong. But Dr. Michel, which strategy would you recommend for proteomic studies? Mass spectrometry, SUMOA, or mesoscale discovery, or other?
So I think that builds nicely off of our last discussion, and that we really need samples that we're comparing across all these different assays. In terms of thinking about clinical use, we'll have to determine what's most feasible for high throughput if it does get at the population level. But the other thing that don't think so much about is that it's not just the platform differences, but sometimes on these different platforms you have different antibodies. And so whether with p-tau, whether it's an N-terminal domain or a mid-terminal domain. And so sometimes it might be difficult to compare the platform when you've actually got two different kind of assays as well. So, right now, we don't know. I think we'll have to see. Results have been shown to be promising regardless of the platform. But again, depending on how it's used and the need for high throughput and other things, direct comparisons are needed.
Thank you. Henrik, maybe you can allude on the difference between different proteomics technologies and which to use for CSF or for plasma analysis.
I think one should go with depending on the research question. or perhaps not depending on the research question, but different proteomic technologies will reveal or visualize different subproteomes that we see over and over and over. So if you have a nice cohort study with a lot of samples, and you have the funding, it would make sense to do some panels and some, if you really want to go broadly and then think carefully about replication, then I think one can go for, Olink panels or Neurological, there are TMT, proteomics and peptidomics data sets, there is lipidomics and metabolomics and all those things. But of course, it's you end up in an extremely advanced statistical analysis, where replication, of course, will be key. But different, I must say, that has come to that's an experience we have had over and over in the lab, that different techniques can make it possible to measure different proteins, and sometimes you don't understand why. So for example, we tried to make an Ebitda oligomer assay, and we thought that Simoa would be good, but then we had problems with bead aggregation and it didn't work out well. And then it turned out that the oligomer assay we eventually developed, even though it's not a great assay, but it worked analytically better on the single molecule counting platform that Merck sells, and that's another that is not bead based in the same way, and you count the detector antibody instead of the bead, the positive beads, and perhaps that was better for a sticky protein. So I would say that one could remain a little bit agnostic to what technology to start with, but try out different technologies and hope for some luck. Of course, you have to do it in a systematic way also, but I mean, one reasonable start, if you have a target, is to start with an ELISA and then transfer it onto Simoa or SMC if needed from an analytical sensitivity point of view. But then omics technologies, there are many to choose among now, it's not easy to say which one to prefer, and it can make sense then to analyze the same sample with multiple technologies to get a broad and deep coverage.
I think it's also just, talking about the differences in terms of antibodies across some of these platforms and kits that are available. But, even with amyloid beta, whether it's, X to 40, X to 42 versus 1 to 40 and 1 to 42, that can make a big difference. And, sometimes it may not be as readily apparent, particularly when you're reading the literature, and that in and of itself can have an effect.
But it's a nice situation to be in, the situation we are in at the moment, that we have biomarkers that can be measured with slightly different technologies, and still we get similar results, and many of the assays correlate, even though the slope is not one. But even though we talk about standardization, and so it's nice when there are some different assays giving the same results. I think the risk of measuring something which is confounded is less when we are in this situation, even though that might mean that we have to at the moment use different cut points since the assays are not calibrated against each other and such things.
Yes, yeah, it's exciting times, and we have lots of possibilities. I do think we still have half an hour, isn't it, for this discussion? So we can continue. Oh, no rush. So I'd like to give the floor to a live question of Dr. Boada. I think that's a nice intermezzo. So can someone open the microphone for Dr. Boada? Or herself? Are you still there, Dr. Boada? Now, maybe we first do another question, and I'll try again in a few minutes. The question of Alicia Nadal. Oh, you're here?
I am here.
Perfect. Now, yes, we do. Welcome.
Thank you very much. Congratulations for all 4 persons that I can see in my screen. Thank you very much, and my gratitude for your time and to collaborate with this research summit. I have a very practical question for Michelle. Okay, because in your final remarks, you are talking about to move towards clinic. okay? And my question is, in terms of biomarker plasma biomarkers, how close we are to usually in the clinical practice, focusing two different scenarios. One, for the clinical trials, could be possible to use plasma biomarkers to include in the criteria, from the criteria inclusion in the clinical biomarkers, because could be very useful and could be more easier or more practical from the lumbar tap. And the other is in terms to the use from the PCP's settings. Could be in the future, nearly future, the plasma biomarkers uses for the PCPs as a screening of AD, or you need more robust specificity, like 100%, because these two scenarios are really very handy into use biomarkers. Okay, thank you very much for offering me the microphone. Okay, my congratulations, of course.
Well, thank you so much for your question. And I think you really hit the nail on the head, where that's the $1 million question that we're trying to understand right now. I think in terms of use for specialty clinics, neurology clinics, I do think that we are closer there. And I do know, as I mentioned, C2N diagnostics, WashUse does use that clinically, especially for individuals that are not able to have lumbar puncture for collection of CSF or amyloid PET. So it really gives an outstanding tool then to be able to try and measure some of these protein markers. I think, from when we first have been thinking about blood-based biomarkers, the whole, or one of the big exclamation points of that was just that you can use it at the primary care level. It's much more feasible, it's less costly, and also in more rural areas. And so that's really been, in terms of either a screening or diagnostic mechanism, something we've been shooting for, and has, historically has been a pipe dream, but I do think it is coming down the road. In terms of how long it's going to be, as I highlighted in my talk, I think there are some important things that need to be considered. So in terms of just understanding these blood markers in the general population and how they can be used. So for example, when I showed you with CKD, that would be pretty much any CKD patient that comes in, or somebody with chronic kidney disease stage, especially 4 or 5, would have higher levels of plasma p-tau. And so as we do understand this, we have to educate the physicians as well, not to diagnose disease, and to understand really what it means. Our sensitivities are fairly low, but the specificities are really high. So, I think going forward, there is a lot of work on developing algorithms with a combination of amyloid and phospho-tau and NFL and perhaps some other markers. And I think that's what's really going to be most beneficial. There are papers that are starting to come out on this, but we need to replicate across a variety of studies. And as was discussed before, considering the percentages of ApoE4 genotype, age, and other things as well. So we're getting closer. It's not going to be next year, but I am really excited about it. And it's been so fun to be in this field for the last 5 years, because the growth has just been fantastic.
And for the happy future about the biomarkers, you talk about 1 year, because that is just in the corner. Thank you very much. Thank you very much.
Thank you very much, Merce, for your question. So we can continue still our nice discussion. Let me see. One question from Jose Vicente Sanchez. Can you distinguish between fast and slow AD progressing patients using either CSF or blood biomarkers? Shoot.
Yes, should I start with CSF? Yeah. On the group level, yes, you can. So if you look at individuals who have who are amyloid positive and have similar cognitive dysfunction, and then you sort them on the basis of whether or not how high the total and phospho-tau increase is. Then you will see that those with the highest total and phospho-tau increase have the fastest progression. If you add in neurofilament light on top of this, it gets even clearer. But I would warn against doing individual risk prediction on the basis of these data, because it's not robust enough. So it's not like one could talk to a patient and say that Ebitda 4240 is positive, but the total tau and phospho-tau are reasonably still not that abnormal. So you could expect a slow disease, or that they are very abnormal and that one should rapidly sell the company or do some personal decisions on the basis of that. I think it's better to go with the symptoms together with the patient than with the biomarker result. I think that when we have had some complaints in relation to the biomarkers, it has been when they have been interpreted like this. For example, a person who he also had a mutation and was amyloid positive, and was advised that he was close to dementia, and changed a lot of important elements of his life to prepare for that, and that it didn't happen, and took 5 more years before the symptoms started to deteriorate. And he was actually very unhappy about that, and not at all. So again, group level data, yes. Individual data, be super careful, and I would actually almost advise against it at the moment.
Michelle, do you have anything to add from your viewpoint of plasma biomarkers? Did you do any studies?
So I think that the plasma biomarkers here are really lagging behind CSF. So we know much more in terms of disease progression and CSF, as Henrik iterated. I guess when we're thinking about disease progression, again, that has been a benefit and thought in terms of blood-based biomarkers, because you can measure them more serially. And so, hopefully down the road, especially if we're looking at a marker of neurodegeneration and rates of neurodegeneration, that might be something that we could use. And I believe is being incorporated in some clinical trials as a secondary endpoint. But right now, we don't have enough information.
Thank you.
I have a point also for Henrik related to this question raised. The people with the I would say incomplete profile, that kind of patients who have only tau positive, only p-tau positive, assuming that these are amyloid related damages, how to interpret these kind of findings? It is necessary a follow-up lumbar puncture for that people, and when in that case?
Yeah, I think follow-up examinations are extremely important. And I mean, that's how we often work in medicine. I think gray zone results where people are close to the cut point, it would make very much sense to invite back, reassess the person, like we often do when we are uncertain as doctors. So I would strongly advise to work like that. And I tell the patient that this is, the picture is not completely black and white here. It's really a little bit in the middle. We are uncertain, and we would like to follow you carefully to make the diagnosis as correct as possible. I think there is no meaning in doing the lumbar puncture too early, and actually when we have looked at 2-year stability, biomarkers are pretty stable. But if you have gray zone values, this could mean that the person is about to change, and the biomarkers might actually be a little bit more dynamic in that range. Also, if you recall this slide I showed on how rapidly people move in their CSF Ebitda 4240 ratio. So I think inviting a person back, of course, the clinical picture will determine how fast you need to do this, and if you have to help the patient and or his or her family by other means. But if you invite the person back and do a new CSF within a year or so, I think that could help to make the picture a little bit clear. And then I'm sure that there will be individuals who remain gray zone and where the data do not fit with the models and explanations we give which is of course often that's also a case that we recognize from other parts of medicine. So that's I really think this type of inviting back for a new examination is good.
And then it's interesting to know whether the blood-based biomarkers could replace the CSF analysis.
Exactly.
And that's also a question asked. So especially here from Alicia Nadal, she asked whether you foresee, I think it's a question for Michelle, do you foresee plasma biomarkers could completely replace CSF biomarkers in the future? Or plasma biomarkers would be used as a kind of preliminary screening or pre-screening, and CSF will always be needed for AD diagnosis or PET?
So yes, I don't see plasma completely replacing CSF or imaging biomarkers. I think, as we talked about earlier, it can potentially be used as a screening mechanism. It could be especially important, or blood-based biomarkers could, for those that have indications to lumbar puncture and to PET. So in those cases, the blood-based biomarker would be extremely helpful. But I think going forward, I mean, there's so much other information that you can get from the CSF markers as well when you're trying to determine specifically what type of dementia it is, that can also be very valuable. So again, in that case, I could see the blood being a potential screen, but then you need to have more confirmation. And particularly, as was discussed before, in terms of Alzheimer's variants with PPA and others, that's where I think the amyloid PET is extremely helpful as well.
Yes, but maybe in the future we will have blood-based biomarkers for all different dementias. Now that it's going so quickly due to technological advances. So being positive about the future, maybe in 10 years or so, we could. But I agree with your point. In CSF biomarkers, there is still a lot of information to be gained. Henrik, do you want to add also to this?
Yeah, no, this positive note you just gave here, Charlotte, I think is good. And it's exciting to think 10 years back also, because then I didn't think we would have, I couldn't imagine that we would have these phospho-tau tests actually, and that they would look so surprisingly good actually. It's so, and then there are so many exciting things happening also in basic biomarker discovery. I was thinking about a paper I just saw, it came last year but I missed it, but that was from Claudio Soto's team working again on this RT-Quick assay to detect seeds of misfolded alpha-synuclein in this case. And then they took it one step further. They did that assay on patients with Parkinson's disease, and then they compared with multiple system atrophy. So they took the samples, they spiked in the recombinant alpha-synuclein, then they isolated the fibrils and did electron microscopy on them and could see that they could detect different types of fibrils. So I started to imagine when I'm reading that paper that if it would be possible to take a blood sample, isolate neuronally derived exosomes, and also in them detect seeds of misfolded alpha-synuclein, look at the end result of the aggregation process, and by that sort of fingerprinting what type of, but from a blood sample. I mean, those, of course, these assays do not exist, but why wouldn't they in some years? So it's
lengthy assay and very complicated.
Yes.
Yeah. So I mean, in that regard, I could see potentially replacing CSF with the blood, but certainly the imaging I think is going to be beneficial just to understand the regional distribution of pathology and atrophy and whatnot.
I agree, and we do have those imaging capacities. So it nails down to brain pathology. It gives an extra confirmation of that. So I do think that imaging is still very important. I have another nice question from Daniel Akolea. He asks whether the immunobest assays, what are your expectations about that? So do you think they might equal or outperform mass spectrometry assay in some way?
Immunoassays
was it for amyloid or is it general?
I do, yeah, amyloid, blood amyloid detection. Yeah, for for tau, we don't have a mass spec yet that's working.
Exactly. No, so I hope they will. I almost said here, but it would be so much easier if we had these types of rapid clinical chemistry automated analysis methods to run on the clinical chemistry instruments. But we don't know if that will come true, and it might be that mass spec. I mean, all the results published so far indicates that mass spec gives you a superior quantification of Ebitda, and especially the Ebitda to 40 ratio. Ebitda is almost made for mass spectrometry. I should add, it's such a nice match. But I am still hopeful. I think we have seen results from some of the larger clinical chemistry companies that they can make assays that measure Ebitda 40 and 42 rather good, and perhaps it will be possible to eventually make them similar to the mass spec based assays. It would be so much easier.
Yeah, sounds good. Yeah, for higher throughput and also the costs. I was shocked, Michelle, for the C2N rates of 1,250. That's much more than the CSF analysis is currently in the lab. And there, for one biomarker, it's about 50 euros. So 2 biomarkers, now it should not be more than 80 euros. So that's quite a difference.
So it is actually in the US, though, from a clinical standpoint for CSF measures, more expensive than the 50 euros that you're talking about. So I think they're, I don't know exactly how they came up with that, but it was based on a lot of different avenues.
I think it is that one counts physician time and that there has to be an anesthesiologist in the house when you do a CSF sampling because of, so it's, but these exercises with calculating costs for examinations, it's very different in different parts of the world. Here in Sweden, with the state-financed healthcare, you can ask for almost anything, and it will not be. You will not get an individual response that you have ordered a too expensive examination. It's quite. It's very different, actually.
Well, and for us, if it depends on if something is covered by Medicare or not, because Medicare will influence and determine what the cost is. Amyloid PET currently isn't covered by Medicare. These plasma measures aren't either, and so then it's a lot more variable.
And going a bit further and deeper into what are we measuring in blood? So what are your hypotheses to explain the changes in amyloid levels in blood are actually reflecting the change in the brain instead of changes in peripheral expression? Who wants to shoot?
So I think one of the, particularly with p-tau, there are pretty good correlations between plasma p-tau and CSF p-tau, which is a strength, as there is with NFL. So I think that certainly is helpful. There's always going to be impacts of peripheral aspects on the biomarkers. One thing I didn't talk about, we've been doing some work with total tau, and it appears that they can be in the adipocytes as well. So body fat potentially could impact total tau levels and things like that. And that's where, again, trying to understand what these look like in the population and factors that might affect them will be important, particularly if we're trying to use these as screening measures where they will be used in a wide distribution.
Could I ask, Michelle, is your feeling also, perhaps I reveal what I feel, but I mean, this, it feels like there are more potential confounders in blood than in CSF, but perhaps that I'm more used to CSF.
No, there are also more proteins and more organs that are in contact with the plasma, and the throughput is maybe higher, shorter. Due to the flooding through the liver and so on. What do you think, Michelle?
No, I completely agree. And that's where, I'm so excited about the blood markers and on these questions in terms of assay. I really leave that to you guys. You have much more expertise on this, but that's really my interest in terms of trying to take this to the population and how do we do that and develop it, the context of use and whatnot. And I think with blood, there is a lot more that needs to be done in terms of fully understanding these.
Yeah.
But then we also have so many wonderful cohorts to do these studies in.
Right.
It's so much richer when thinking about blood samples and big cohorts, and also the possibilities of looking at genetics and lifestyle-related factors. This also with the polygenic risk scores and risk and resilience. And I mean, to be able in your cohorts, Michelle, to look at people with high polygenic risk who do not get biomarker abnormalities, and those with low genetic risks who still get it. What are different between those? And it must be wonderful to start PhD student projects on such in such studies. I mean, there must be a lot of things to learn from such studies.
Yeah, there is. But I think that, Charlotte, with what you've presented in terms of standardization too, that the one really good thing that allows us to look at all these cohort studies is that, freeze-thaw cycles, they can go through a couple for the most part, and it's not, having to put them in -80 right away. So, I mean, that's been really beneficial in determining, yes, we can go forward with all these stored samples that have been collected.
Exactly. Yeah. So, and that's remarkable. Who wanted to say?
No, I was mentioning also the PRS incorporation into the modeling of the risk, but even beyond the polygenic risk score associated to the disease, which is clearly important to be taken into account. Also, the SNPs and the variation in the genome also affecting to the natural levels of the proteins we are measuring as well. So probably we have to incorporate also these variants in the general population related to the genetic variants behind to increase the accuracy of the methods. Do you think that is also feasible?
Well, in terms of cost, because then you have to do this extra genetic analysis also. So if from a very practical standpoint, I think that's not so easy. But if it's needed, I don't know what the others think about it.
I think it's an exciting idea, and I think also in the larger cohorts where we have genetic information together with biomarker data, we might find new mutations, for example, or new genetic. I'm thinking a little bit about this Christchurch mutation that was found in the Colombian kindred, biomarker discordance that made the researchers sequence that individual, finding a new ID. I think there are interesting things also with the blood Ebitda 38, 40, 42 ratios. There, I think if one looked at that in large studies, one would detect novel presenilin mutations, for example. So I think there are a lot of exciting interactions between genotypes and fluid biomarkers that we could, where we could find new things.
Yeah, so it's very interesting. Michelle?
Oh, I was just going to say, I know we've been contacted by, I think, 3 or 4 groups now who are trying to put these, have large consortiums where they are looking at total tau and NFL and p-tau. And so hopefully we'll have some answers or guidance on that soon.
It's still a bit challenging in the lab to do 20,000 biomarker measurements or 50,000 or 100,000. Then we would need to increase our capacity.
Big databases. But nowadays that's also possible with big servers also. There are still many interesting questions. What's the effect, for example, of the platelet-derived amyloids? Any ideas on that? On the plasma amyloid levels, of course.
I don't, Henrik or Charlotte, do you know? I don't know.
We have done some studies where we tried to address this, but we didn't get wiser, unfortunately. So for example, you can prepare plasma by centrifugation at different speeds to create platelet-depleted or plasma. And we hoped that we would get a better Ebitda signal if we removed platelets, so that we wouldn't have a background level of platelet-derived Ebitda in our measurements, but there was no big difference actually. So we have not. It's a very interesting question. Everything that deals with peripheral Ebitda, if one could solve that problem, that would be a great thing for the plasma Ebitda-42/40 ratio, because I think this is the explanation, at least a part of the explanation for why Ebitda-42/40 ratio has a slower percentage decline in amyloid positive individuals, because we have this background Ebitda level which is not affected by the pathology. So it's an important question. Someone told me that myocytes also produce quite a bit of Ebitda, and but otherwise platelets, of course, are known sources.
But are platelets not activated upon clotting? So that would mean that there will be amyloid released from platelets if you have serum, but we don't see a high amyloid level in serum samples. The contrary, we see lower levels in serum compared to the plasma. And that accounts for nearly all assays that we tested. So, based on those results, I'm less convinced of the contribution of the platelets to what we measure.
But don't you think that Ebitda ends up in the clot?
Oh, that's also possible. That's possible. But then the effect of the platelet release is then mitigated by that. That's what you think.
Yeah, but it could also, of course, bring down Ebitda.
So those effects together end up in a lower level then. Yeah, could be. Good idea. Yeah.
But if that was the case, perhaps the ratio would be a little bit better in serum, even though the absolute levels would be lower. Have you tried that?
I think the ratio was a bit better, but need to go back. But often we can't even measure it well in serum, the amyloid beta 42 at least. Yeah. Okay. The ratio was always better, whatever you do.
Always better, yes.
There's a question. How are we going to establish cutoff points in asymptomatic individuals? What's the golden standard, where it points down to? Using a PET scan, or is the meaning of a positive PET scan the same in cognitively healthy compared to symptomatic individuals? Oh, now we're going into the PET verb. Who is familiar or comfortable with that?
So all the biomarkers and PET itself is a biomarker, has room for error. And so I think neuropathology in general is kind of the gold standard. The problem is, of course, that you have to wait. There's typically a lag between having the blood draw and then having autopsy samples. So, in terms of establishing the cut points, I think the best thing to do would be to look at it in relation to autopsy, in relation to amyloid PET, and try and understand what some of the differences are.
Mm-hmm.
I could just say that I think the molecular imaging has helped us a lot with the blood biomarkers, because it is so clear that before when we also had this problem that 30% of controls had some amyloid, it was very hard to see anything. But now for the Ebitda-42/40 ratio, I don't think we would have been able to establish the difference without amyloid PET.
Okay.
I definitely agree. But I just recently got a response back from a reviewer, so maybe it was one of you guys, but with pointing out that neuropathology is the gold standard, and so using amyloid PET is a limitation. So that's where that background is coming from.
I was not a reviewer, no.
But the different forms of amyloid, I agree. Cord, plaques, diffuse plaque distribution, tissue reactions around them, a lot of things can probably be, a lot of additional information can be gained from neuropathology.
I think we managed to go across almost all the questions, or maybe even all. So I think we can round up now. And so I give the floor to Augustin to give the last words.
Thank you, Charlotte, and also to all of you for these very, very amazing discussions and your presentations. I think that we will need another hour, even more, to speak about bimodal distribution and many other stuff. Very, very interesting you show in these developments. But I have to close the session. And I wanted to thank Professor Teunissen for sharing the session, of course, and also to our partners, Grifols and Pharma, the audience for lively discussion, and of course for Michelle and also for her representing this amazing presentation. I'd like to invite all the people to join for the next month for the third session of the ACE Global Research Summit 2021, which is going to be held on June 25th. I will focus on blood circulating factors contributing to dementia pathophysiology, and will be hosted by my mentor and my colleague, Professor Mercè Boada. Please also help us to improve the session by taking a few minutes to fulfill the survey that we will come up to your screen in a few seconds. Thanks again, everybody, and we hope to meet you very soon for another research afternoon. Bye-bye.
Thank you. Bye-bye.
Oh, nice audience also.
Yeah.
Bye-bye.
Bye.